Assalamualaikum and good morning to everyone. We are from Group 12 and we will present about genome sequencing project. So this is a table of contents. I will start with the introduction. The title of the article that we choose is Population Analysis of the Korean Native Duck Using Whole Genome Sequencing Data, and that is the researcher. So this article is about Korean native duck that we call as a woolly. The duck is a domesticated duck originated from the hybridized duck between Malay duck and indigenous Peking duck. It has purpose on economic efficiency and productivity. It also has high crude protein, water retention capacity, and unique meat flavor and texture with high fatty acid. So, from this article, the researchers came with the problem that is, although some recent studies have investigated the unique characteristic of Korean native duck, but by using whole genome level studies of the Korean native duck still lag behind than the other duck. So they came with the solution. They applied a population level genome analysis based on the whole genome sequencing data from the population of various duck breed. This is including the Korean native duck. So they sequenced the whole genome of 20 Korean duck and 14 data from diverse duck breed. Then they discovered the single nucleotide polymorphism for 15 duck breed population, including Korean duck. Then they also identified the genomic region under high differentiation among that duck breed and performed functional analysis of the gene. This method will be explained more later by my friend. So, the objective from this article is to perform population and functional analysis of Korean native duck, which has a distinct meat flavor and texture phenotype by using whole genome sequencing data. And the other objective is to study the distinct genomic features of Korean native duck by conduct population level genomic analysis by using 20 Korean native duck together with 15 other duck breeds.
Here are the methodology used for this genome sequencing project. The first one is sequencing and library preparation. A whole genome resequencing data from a population of 20 Korean native duck was generated. The samples are prepared following the Illumina protocols, and the sample is size selected, aiming for 400 to 500 base pair products in which later then amplified using PCR. Second is read alignment and variant calling. Public sequencing data of various breeds of duck was collected from the NCBI SRA database to generate single nucleotide polymorphism SNP data. A total of 103 public sequencing data of various duck breeds and 20 resequencing data of Korean ducks were aligned to the chromosome level duck reference genome. Duplicate reads which map to the same position on the reference genome are filtered, and local realignment was performed using the genome analysis toolkit. Next is SNP annotation, KD-specific gene identification, and functional analysis. A database was built with the NCBI RefSec gene annotation data of the reference duck assembly. Variant annotation was also performed for the final SNPs of 15 duck breeds. Then the transition to transversion ratio are calculated to evaluate the quality of the SNP. Finally, is nucleotide diversity, population structure, and selective sweep analysis. SNP data was filtered following certain criteria. For each duck breed, the nucleotide diversity was calculated for each of 40 kb sliding genomic windows using the filtered SNPs. A phylogenetic tree was constructed based on the SNP filtered by Pling. To investigate differentiated regions among the various duck populations, the mean FST value was calculated. And finally, the results of the population differentiation were visualized in the form of a Manhattan plot.
Results: the researchers carry out a whole genome resequencing on 20 Korean native duck breeds. In this research, the researchers detected single nucleotide polymorphism (SNP) in 15 different duck breeds, including the Korean native duck breed. SNP is a substitution of a single nucleotide at the specific position in the genome. Around 50 million SNPs were found in the Korean native duck, which is considered one of the highest among the other breeds. Then, the transition to transversion ratio is calculated to assess the overall quality of SNP. A ratio of 2.53 is obtained for the native Korean duck breed, which indicates transition mutation. Researchers noted that all SNP with 19 categories, including synonymous, non-synonymous, and intergenic. The research investigated on the unique genomic characteristic of the Korean duck and found out that 3,062 unique genes, which consist of non-synonymous SNP, which were observed in the Korean duck, were not found in other breeds. at the same position. Gene ontology analysis were carried out to determine the function of those unique genes. And those unique genes were involved in various important biological processes. Among those 3062 genes, there were two genes which were PNPLA8 and INO1, which have different amino acid when compared to other dark breeds, which is due to the missense variant.
Hi, I'm Nishan. I'm going to present the nucleotide diversity, population structure, and the phylogenetic relationship analysis. The mean nucleotide diversity for each of the 15 dark breeds is calculated by the filter SNPs. Two approaches are then used to identify the population structure of the 15 dark breeds. These are the mean nucleotide diversity of the 15 dark breeds. First, the principal component analysis is conducted to identify the genomic relationship among the 15 dark breeds. It is divided into three major clusters by the first two principal components. KD and PK breeds were very tightly clustered together, whereas the MD breed was loosely stretched. The remaining cluster included all the indigenous breeds. Second, the population genomic structure is analyzed. At k equals to 5, the KD and PK were separated and a subdivision in MD is observed. Genomic relationship among the 15 dark breeds observed at k equals to 5 is consistent with the result of PCA. A maximum likelihood tree is constructed to identify the phylogenetic relationships among the 15 dark breeds. It is confirmed that most of the individuals in the same breeds were grouped into one cluster. This result were also consistent with the PCA result in terms of the first two principal components. Next, the population differentiation analysis. The population differentiation among the KD, PK, and MD populations is investigated based on the estimated tree topology. This is the Manhattan plot of the Z transform FST between the Korean native dark and Peking, which is the top panel, and between the Korean native dark and the Malay dark at the bottom panel. In KD versus PK population, some highly differentiated regions included the MITF and B3GALT1 genes. Meanwhile, in KD versus MD population, the MTNR1A and the ITPR2 genes were observed in highly differentiated regions.
Hi, I'm Azra Inshira binti Suhaimi, A169938, and I will continue on the discussion from this article. Based on the results, they have detected a total of 51,154,530 high-quality SNPs from the 15 dark breeds. They have also found 3,062 KD genes containing non-synonymous SNPs and further filtered the KD genes and obtained 103 KD specific genes. Among the 3,062 KD genes, PNPLS8 gene and ENO1 gene are found. Although no significant function has been identified among the 103 KD specific genes, these genes may underlie a difference between KD and other breeds, for example, the GRIK2 genes. The functions of the genes are as mentioned in the slide. Next, we move on to the mean nucleotide diversity of dark breeds, and it was between 0.1028 to 0.1384, and MD or mallet dark showed higher nucleotide diversity than the domesticated breeds. KD has a higher nucleotide diversity than PK, which may be because the breeding history of KD is shorter than PK. They have also performed population analysis of 15 dark breeds including KD and found that KD breed has a close genomic relationship and shared admixture history with the PK breed. This can be explained by the domestication history of KD, which was formed with hybridized ducks of indigenous PK and wild mallet dark in the late 1990s and has been improved to current KD population. A differentiation analysis was also conducted to discover what makes the difference among KD, PK, and MD breeds, and they have identified several candidate regions covering highly differentiated SNPs with respect to KD versus PK and KD versus MD. In the highly differentiated regions between KD and PK, they found several phenotype-related genes such as B3GALT1, FHIT, and MITF. Their functions are as follows. In highly differentiated regions between KD and MD, they found two interesting genes, MTNR1A and IPTR2. Their functions are as follows. They have suggested that these genes in the highly differentiated regions could be candidates for improving reproductivity, meat quality, and egg quality. They have also investigated the highly differentiated regions among KD and other 14 dark breeds and found 53 genes including ABHD17A, SSH2, and TGFB3, and their functions are as shown in the slide. They stated that the genes related with these functions may have created difference between KD and other 14 dark breeds. That's all, thank you.
Lastly, the conclusion is We can conclude that the analysis of current native dog population and functioning can be done by using the whole genome sequencing data. And this finding also help us to know more about genomic characteristic of current native dog and the other breeds. Thank you. That's all from us.